Today we're going to be talking about organelles. And organelles are structures that carry out specific functions in a cell. An organelle to a cell is like an organ is to a body. So let's go ahead and talk about organelles. We're going to be talking about a nucleus, mitochondria, and a chloroplast. Let's start with the nucleus. We're going to go ahead and draw the nucleus. The nucleus is a double membrane structure, and they tend to be round. So there's the outer nuclear membrane. So we'll write that down, outer nuclear membrane. And inside is an inner nuclear membrane. Between the two is an intermembrane space. Altogether, they're referred to as the nuclear envelope. Now, within this nuclear envelope, we have transport proteins. And these transport proteins span the entire width of the nuclear envelope. And there's also other proteins embedded in the nuclear envelope, but these are the ones that we're going to talk about today. So let's go ahead and give them a name. These are transport proteins. And in the nucleus, the transport protein has a special name. We're going to call it a nuclear pore. And the nuclear pores transport polar materials into and out of the nucleus. Nonpolar materials simply diffuse right through the cell membrane. Now, inside the nucleus, we have a series of proteins that actually line the inside of the nucleus. And these proteins act as a sort of skeleton, a protein skeleton. And these proteins provide support. And they have a special name called the nuclear lamina. Their job is support, but they also help out in cell division and DNA replication. Inside the nucleus is also an area that stains very dark. And the reason it stains very dark is because it contains lots and lots of ribosomes, small proteins that make other proteins. When we want to see the parts of a cell, we stain the cell. And when we stain the nucleus, this area right here, and that's simply what it is, it's an area, stains very, very dark because there are so many ribosomes being made there. It turns out that this area contains the DNA that has the information needed to make the ribosomes. So we're constantly making lots of ribosomes that stain a very deep, dark color. So let's go ahead and give that a name. We're going to call that the nucleolus. And its job is to make ribosomes. And ribosomes are small proteins that make other proteins. These ribosomes, after they're formed, get transported out through the nuclear pore and into the cytoplasm where they actually perform their function. Also in here is DNA. And the DNA is actually wrapped around proteins. Let's go ahead and enlarge this DNA molecule. So we're going to enlarge it. It's called a chromosome. And in a human cell, we have 46 chromosomes. 23 we're going to get from mom originally, and 23 we're going to get from dad originally. Now, let's go ahead and enlarge this. If I look at this, there's actually some proteins in there also besides the DNA molecule. I'm going to draw 4 proteins. These proteins are going to be referred to as histones. And behind these 4 proteins are 4 more proteins. So there's actually 8 histones altogether. That's just one group. I'm going to draw another group over here. And one more group over here. And so on, and so on, and so on, until we get to the end of that DNA molecule. So here's our DNA molecule. It wraps 1, 2.5 times. 1, 2.5 times. 1, 2.5 times. Let's label that DNA molecule. And the whole complex altogether is referred to as a chromosome. A chromosome is simply a long piece of DNA wrapped around histones. And it's wrapped around those histones for a reason. Those histones supply support for the DNA. DNA is extremely long and extremely thin. If we were to actually hold it up like a piece of thread, you wouldn't even be able to see it. In fact, if you just blew it, it would probably break in two. And we don't want that. That would create a mutation, which would result in the death of our cell. So we need to strengthen that DNA. And we're going to strengthen it by wrapping it around proteins over and over and over. So it strengthens the DNA. In addition to that, we don't want a tangled mass of DNA. So the protein, the histones, are also going to organize the DNA. So organized. And finally, by wrapping the DNA either loosely or tightly around the histones, we can either grant access to that information or prevent access to that information. So this is a way of regulating DNA expression. Since DNA contains the information that need to make proteins, we don't always want to make proteins all the time. Sometimes we want to make this protein, sometimes we want to make that protein. And we want to make those proteins in certain quantities. By allowing access or restricting access to that information, we can regulate how much and what type of proteins are actually made. So let's go ahead and write DNA expression. Now back up to our nucleus, there's a jelly-like material inside our nucleus, and that jelly-like material is referred to as the nucleoplasm. It's mainly a watery substance with proteins dissolved in it. And by dissolving different proteins in it, we give it a different function. We give it a specific function. And that's going to be it for our nucleus. The next organelle we're going to talk about is referred to as a mitochondria. So let's go ahead and draw our mitochondria. I'm going to write mitochondria up here. And the mitochondria has the function of taking the energy stored in glucose and transferring that energy to the energy stored in ATP. So let's go ahead and write that down. Energy stored in glucose to energy stored in ATP. And where is the energy actually stored? Well, the energy is stored in chemical bonds. The chemical bonds of glucose. Specifically, we're concerned about the chemical bonds, the covalent bonds, between carbon and hydrogen. We're going to break those chemical bonds, release the energy, and take that energy and create new chemical bonds in ATP and store it there. Because ATP is the currency on which the cell operates. That's the only type of energy that the cell can use directly. So all the energy has to be converted to the energy of ATP. Let's go ahead and draw mitochondria. They tend to be sort of oval-shaped. Sometimes they're round, sometimes they're cigar-shaped. They come in different shapes and sizes. We're going to call this the outer mitochondrial, and I'm going to abbreviate that, MIT, membrane. On the inside, we have an inner mitochondrial membrane. So, like the nucleus, it's also a double membrane structure. However, the membrane here is very convoluted. And I'm going to draw it very twisty and windy, like the name implies, convoluted. So, inner mitochondrial membrane. And it's going to be very convoluted, twisty or windy. And that's going to be extremely important. We talked about form and function. A structure has to look the way it does in order to perform its function properly. This inner mitochondrial membrane has to look very convoluted. By being very convoluted, it has an extremely high surface area. And that's going to be very important. The more surface area we have, the more cellular respiratory proteins we can actually embed into that membrane. Now these proteins are responsible for doing this job up here, for taking the energy stored in glucose and converting it to the energy stored in ATP. And the more of these proteins I can embed into that inner mitochondrial membrane, the more ATP I can make. So lots of surface area is a good thing. Between the two membranes is an intermembrane space. And within that intermembrane space are lots and lots and lots of hydrogen ions. So we'll draw a few of them. Now these hydrogen ions represent potential energy. In order to create potential energy, we want to pump lots and lots of hydrogen ions into a small space. That way we can create a high concentration relatively easily. And that high concentration of hydrogen ions is a way of storing energy or potential energy. Later we're going to use that energy to make ATP. Within the center of this mitochondria is a jelly-like material. This jelly-like material is going to be called the mitochondrial matrix. Again, it's a watery liquid containing lots of dissolved proteins. It contains different dissolved proteins than the nucleoplasm of our nucleus. In this way, it has a different function. That's going to be it for our mitochondria.